In this section, I'm going to talk about microbial nutrition and microbial relationships. So, nutrition. Nutrients are acquired from the environment and used for cellular activities. An essential nutrient is a nutrient that cannot be made from that organism. It must be provided by another organism or from the environment. Macronutrients are required in large quantities, while micronutrients are required in small quantities. An example of a macronutrient would be carbohydrates. An example of a micronutrient would be something like iron. Microbes need all of the same types of elements as we do, supplied in their environment. So they're going to need carbon, hydrogen, nitrogen, and so forth. They are going to use this for all of their cellular processes. Microbes also have particular feeding mechanisms. Phototrophs are microbes that photosynthesize. So photo means light, troph is kind of like eating. Think of a food trough. So they're going to eat light, which doesn't really make sense. They use light in order to produce their own food. Chemotroph, they gain energy from chemical compounds. So they need chemicals in order to gain energy. An autotroph makes their own carbon source, while a heterotroph has to obtain their carbon source from outside of the cell, either from the environment or from another organism. So this is an example of a bunch of different types of nutrient categories of microbes by energy and carbon source. So a photoautotroph is going to make its own carbon using energy from the sun. That's photosynthesis. So photosynthetic organisms such as algae, plants, and cyanobacteria. A chemoautotroph is going to make its own sugar using organic compounds. A chemoautotroph with lithotrophs, chemolithoautotroph, is going to make its own carbon source using inorganic compounds. A photoheterotroph is going to use light for energy and then eat its carbon source. A chemoheterotroph is going to eat other organisms or eat their carbon source from outside, and they use chemicals in order to do that. A parasite is going to utilize the tissues or fluids of a live host. So chemoorganic autotroph uses organic compounds for energy and inorganic compounds as a carbon source, while chemolithoautotrophs require neither sunlight nor organic nutrients and rely totally on inorganic materials. Chemoheterotrophs, so a majority of heterotrophic microorganisms, people are also chemoheterotrophs. They derive both carbon and energy from organic compounds. Organic molecules are processed through respiration or fermentation, and they release energy in the form of ATP. Saprobes are going to decompose plants, animals, dead microbes, and they're important in recycling nutrients. Most saprobes have a rigid cell wall, and they cannot engulf large particles, so they're going to release digestive enzymes, digest those materials externally, and then absorb just the nutrients. Bacteria and fungi are saprophytic, or can be saprophytic. So now that we've talked about those nutritional categories, we're going to talk about some associations between organisms. Symbiotic means that organisms are living in close nutritional relationships that are required by one or both members. Non-symbiotic are organisms that are free-living and their relationships are not required for survival. Examples of symbiotic relationships include mutualism, where both organisms benefit, commensalism, where the commensal benefits and the other member is not harmed, and parasitism. Parasite is where one benefits and the other is harmed. If we think about mutualism out in the environment, where both are going to benefit, we can think of the root nodules in plants. They're a nitrogen-fixing bacteria that live in a mutualistic relationship with plants. The plants grow these special root nodules which house this nitrogen-fixing bacteria. Nitrogen-fixing bacteria produce a nitrogen compound that can be used by the plant to make amino acids. In exchange for producing that, the plant provides that bacteria a safe house to live in. So protection, comfort, and additional nutrients from the plant. So they live in a mutualistic relationship. Commensalism. The commensal benefits and the other member is not harmed. This would be, for example, a microbe that lives on the surface of a fly. So those microbes are getting protection, they're getting to eat nutrients, but they're not necessarily harming the fly. The fly doesn't care that they're there. Parasitism is where one benefits and the other is harmed. So here we can talk about when we get an infection. The microorganism is benefiting because they're eating our nutrients of our body, and we are being harmed in the process. Non-symbiotic relationships, like I mentioned, they're organisms that are free living. They are not required, their relationships are not required for the survival of the organism. So synergism, it's when members cooperate and share nutrients. And antagonism is when members are inhibited or destroyed by others. So this relationship isn't necessary for anyone's survival, but it's present. So they happen to either feed together and share, or they fight against each other. So these definitions kind of go into this in a little bit more detail. So symbiosis, a general term that's used to denote a situation in which two organisms live together in a close partnership. Mutualism, it's when they are obligatory and mutually beneficial. Commensalism, like I said, one benefits, the other one is unaffected. And parasitism, one benefits and the other is harmed. Antagonism happens during competition. Synergism is when they both kind of get along, but their relationship is not necessary for their survival. Biofilms, as I talked about a little bit earlier, biofilms is an example of synergy. So they're all living together, they're all getting along, they're sharing things, and they're also communicating with one another. And the way that microorganisms communicate with one another is usually by chemical communication. So they're going to spray out some chemicals, get chemical signals back, and they can start to form these complex associations. They talk to each other by quorum sensing. Quorum sensing allows the biofilm organisms to determine the size of the biofilm, to determine how many other organisms are present, and that determines how that microorganism is going to interact with its environment. So, if I'm a bacterial cell and my special power is to produce light, so I can glow in the dark. And there are bacteria out there. We'll learn about those bacteria when we talk about bacterial genetics. But now let's say that I'm living inside of a squid, and I want to start producing this glowing protein inside of a squid because I want to make this squid glow in the dark. If it's just me, as a bacterial cell, I'm not going to go through all of that energy and all that work to produce this glowing protein if it's just me. Because in the big scheme of things, it's not going to matter. It's not going to be seen by any organisms. But if I do quorum sensing, if I spit out chemicals and wait for a chemical signal back, and I'm sensing there's a lot of organisms like me around me, then I'm going to start producing that protein. And those organisms are also going to start producing that protein. And before you know it, we're all going to be glowing. And you're going to be able to see this glowing because you're going to see that there's a lot of us glowing together. So that's how quorum sensing can help change the behavior of certain organisms. You determine which organisms are around you in order to produce or not produce certain metabolic products or certain enzymes, certain proteins that are going to have some effect on the environment.